Okay, thank you very much. I'm going to give a 25-minute talk now on COVID-19, aspects to do with many different activities that are happening in the world today around this virus. And I thought I'd just start with this next slide here. Same as it ever was. This is 1918 flu pandemic. Guess what we were doing back then? I will mention this as well, I'm sure. Do not disregard the advice of a specialist just because you don't understand. Do not disregard the rights of a community. And look at this, wear a mask, lead a clean, healthy life, wash your hands. I mean, it's the same thing. Who would have thought all these years later we're still doing exactly the same thing? You don't need much science actually at the moment if you look at the world when it comes to how we're handling COVID-19. All these measures were in place in 1918. Of course, we hope that science will eventually save us from this virus. Now, quick bit of background. You all know the virus itself is called SARS-CoV-2, obviously. It's still astounding to think that these numbers, we'll talk about these for decades to come, that a third of the world's population was put into quarantine because of this virus. 1.2 billion children were kept out of school. Trillions, that's an estimate, 100 trillion wiped off the world economy, and many other consequences of this virus. Nothing like it, of course, since the 1918 pandemic. This has many features, of course, that make it different to that in all kinds of ways, but still an alarming turn of events in many ways. Now, what the virus is, of course, you all know lots of this. That's what it looks like. I think Peter Medawar, one of the world's most famous immunologists, actually, he won the Nobel Prize for work on transplantation. He called the virus a piece of bad news wrapped up in a protein. That's exactly what it is. In this case, it's got spike proteins around the outside, membrane protein as well, and then there's the RNA molecule in the middle. That's the recipe to make more virus. We know, of course, it's a coronavirus, and there are other members of this family. This is quite a good diagram I got, I think it's from Science magazine actually, where, the other coronaviruses, SARS and MERS, this is the time they think you can get protection if you're infected. So in other words, if you have SARS or MERS, there's evidence of T cells persisting for 2 years even, 34 months in the case of MERS. In the case of the ones that cause the common cold, there's the four of them there, 6 months protection max would seem to be the case. And then in the case of this one, of course, we just don't know. And it's a big worry at the moment that if you get infected, you probably will get reinfected. And we think at the moment there'll be a kind of a normal distribution of reinfection actually, like most biological traits, where some will get reinfected for definite, some will be protected, but the majority will probably get reinfected with mild disease, as happens with the common cold. This is a more when it comes to vaccines, though, as I'll explain a bit later. But the fact is, this notion of herd immunity then happening naturally is wrong. And no immunologist that I know of is suggesting that because it's too dangerous. The reason being reinfection is a reality. And that means that the virus just keeps persisting in the population, becomes endemic. And of course can spread then to vulnerable people and harm them. So this notion is really not seen as viable. The ones who advocate for herd immunity are epidemiologists largely, as far as I can make out. And then of course there's flu and there's measles as a comparison. 6 months protection from flu, lifelong with measles. So that's better, obviously, than these other ones. But still, SARS-CoV-2 is still not fully clear. And then this is an important recent fact that we must keep reminding people this is not like the flu. Now, what's the evidence for this? This is a great study was done, actually. The reference is at the bottom of this slide, I hope. This is comparing the infection fatality rate based on age. Now, look at this. Blue is influenza. Red is COVID-19. Lots of different studies. Now, look at these numbers. If you're 40, you have a 5-fold increased risk of death from this virus. If you're in your 70s and 80s, it goes up to 13-fold increased risk of death from this virus when compared to flu. So people should stop saying it's just like flu. There's the scientific evidence. And now if extra evidence comes along to deny this, we'll look at it. But certainly all these studies are indicating strongly it's a much more severe disease than flu based on age. If you're under 40, of course, and under 30, it's around about the same. These overlap a lot down here. But still, as you go beyond that age, it's much more dangerous than flu. Now, of course, it came from, as you all know by now, bats. The closest relative is in a bat, and there we have the relationship. Also in the pangolin, so it could be pangolin derived. The ancestor may go back to the year 1200 based on genomics. So these coronaviruses have been bumping around for a long time, and eventually SARS-CoV-2 got to us. Lots of work is going into, of course, the origin of these using RNA analysis and genomics to suss this out to try to prevent the next one happening, I suppose, is the idea there. The thing to mention that, again, you will all know by now, you couldn't wish for a worse virus in some ways. I mean, obviously the 1918 pandemic was vicious because it killed lots of young people. But the way this one is designed is especially devious and pernicious. It's because it's a bit like the ones that cause the common cold on the left here. and the ones that cause SARS and MERS had a baby, and it's called SARS-CoV-2. What do I mean by that? Well, it can live in the upper airways, just like a cold, very limited symptoms, but can spread from there, or it goes to your lungs, like SARS does, and then you get really sick and you spread it by being sick. This one can live in either place, which is the reason for asymptomatic spread, a really nasty aspect of this virus. It couldn't be shut down then because of asymptomatic spread, unlike SARS and MERS, which could be shut down because that was symptomatic spread. You could just quarantine people who had symptoms. In this case, many people walk around with no symptoms, and it's up in their upper airways, and they keep spreading it, and it hits people, and some of them die because it goes into their lungs. Now, again, this is like the flu slide. Obviously, it's a disease of older people. And here's the infection, 4 separate studies here. As you get older, the IFR goes up a lot. So we know it's a disease of older people. Young people aren't spared, though. Remember, there's still some serious consequences there, and they can be a source of spread as well. And then a very interesting aspect that's becoming clearer. Another great the 2 big unknowns actually are, can you get reinfected? And I just mentioned that a few minutes ago. Evidence is you can. The second big unknown is long COVID or persistent symptoms, and this is called long COVID. It can affect concentration, shortness of breath, the heart, hypertension. All these things are a feature for months after infection, and we're still trying to get a handle on this mechanistically. Is this some kind of autoimmune consequence of infection is one idea. We don't think the virus goes latent, so I can't really explain this. It's a big unknown. Lots of research happening here. If we can find the basis for this, of course, this could inform any post-viral fatigue syndrome or any post-infectious symptomology, which is a reality from any infection. So it's a very important area of research, really. Now, let's move on to the hopes and the kind of targets, I guess, and how we're going to get at this. We know an awful lot about this virus now. There's something like 60,000 papers now in PubMed if you search for SARS-CoV-2 and COVID-19. And like other coronaviruses, like SARS and MERS, ACE2 is the receptor. Into the cytosol, the RNA gets released, starts replicating, and you make viral proteins as well, and then you package the virus and then you release it. So it's quite straightforward as a coronavirus goes. And obviously the immune system has to kick in. As the RNA levels plummet, antibody levels go up, IgM and IgG, of course. The big question is, can this persist? We've no doubt now there will be memory B cells will come out. You mightn't measure antibody in those people because it has fallen. But the hope is there's some antibodies. But again, the unanswered question, would they be sufficient to confer protection from reinfection? There's also a worry of a thing called AID, antibody-dependent enhancement. With other viruses, you get that the antibodies coat the virus and enhances uptake into cells. Is that happening with SARS-CoV-2? We still don't know. It's a big unknown that may have relevance for the vaccine as well. We also know a lot about the T-cell effect, and there's no doubt you get T-cell responses, of course. You need a strong CD8 response to get rid of viruses, remember. What does this virus do? It decreases the T-cell count and causes a lymphopenia. And there we see peripheral blood T-cell counts plummeting as the viral load goes up. This has 2 consequences. The virus now goes really rampant, driving inflammation. You're losing negative feedback loops actually on the inflammatory process by getting rid of regulatory T cells, for instance. There was also a study last week which suggests the germinal center where B cells go and the lymph nodes starts getting damaged as well. So it's a very pernicious virus in terms of how it modulates our immune system. Now, how are we going to fight this? Well, obviously it's a battle on several fronts. Clearly, test, trace, isolate is the first front. We've lost that battle in Ireland, and I can't see it coming back. The Chinese beat it through test, trace, isolate. Can you believe the Chinese have tested 9 million people in 2 days? That's twice the population of Ireland. Why we can't do that here is still a mystery to many of us. The second method is the medieval method of distancing, hand washing, all that sort of stuff, wearing a mask. This is where science comes in. Will there be a vaccine? Antibody-based therapeutics, direct antiviral drugs and anti-inflammatories. Now, I'll just go through these one by one for you very quickly. This is the Wellcome Trusted, a very nice slide. That was from my kids' book, actually, which I've just done a reprint of. But this is the Wellcome Trust fancier version. Here's the antivirals that stop the virus in the body. Remdesivir is the example there. Anti-inflammatories, that's my area, on that towards the end. And antibodies, which can be used then as treatments. And there's a big hope with those. There's 44 in trials at the moment. And Eli Lilly have one, Regeneron have one, of course. They have a cocktail of antibodies. And that's obvious. It'll just mask the spike and stop the virus from getting inside cells. All of these are underway. I'd be most optimistic about the antibody treatments. Although the Lilly trial, I just noticed 20 minutes ago, was halted because of an adverse event. That happens in trials. It wouldn't perturb us at this stage. But still, antibody approaches, I think, could be very promising indeed. And now the notion of the vaccine, remember, and herd immunity. Again, that's plugging my book again. That's the kids' book. I've got a section on immunology in it. Herd immunity is what it's all about. Now, remember, we don't think it's possible to get this to any great extent through the natural course of infection. And here's the reason why. Here's a natural infection with no immunisation. Let's say there's no distancing, spreads like wildfire in the population and many people die. If we have social distancing, of course, we slow that down and the R number drops, less deaths are happening in this one here. But look at this, immunisation. You will have 1 or 2 cases, but now we protect the population. So you need to immunise really to achieve herd immunity. And social distancing remains a key strategy anyway, just to re-emphasise that. Now, update on the vaccines. It's alarming and compelling and so exciting if you're into vaccine development and if you're into immunology, because there's an absolute frenzy of vaccines being tested. 11 have now got to phase 3, for example. We think this includes some of the Chinese and Russian vaccines. There's probably over 200 in development at the moment. And here are some of them. There's different types of vaccines, as I would guess you all might know. Hodin activated is the standard way to vaccinate. Sinopharm and Sinovac have that. Using subunits, the S protein, the spike, Novavax, Sanofi, GSK. Using nucleic acids, there's two RNA vaccines, Moderna and the Pfizer BioNTech. That should be a B there. And then the recombinant viral vectors, that's using adenoviral vectors to deliver the spike protein, usually in these vaccines. That's CanSino, the AstraZeneca Oxford vaccine, and J&J. So all of these are different strategies. It's good there's so many strategies because one might work, another one won't. It gives you more shots on goal, I guess, is the idea, using these different strategies. And just today, it's Tuesday today, Barclays Investment Bank have been very bold in more ways than one, probably. They've done a projection as to when the vaccines are going to be available. They're projecting that towards the end of this year, Moderna, Pfizer and AstraZeneca vaccines will be approved. Isn't that staggering? And then as we get to the middle of next year, then the other ones, and J&J, Novavax and so on. So is this true? Well, they're optimistic. We'll see. I've no doubt there will be results from these trials towards the end of the year. And let's keep our fingers crossed. Even if it's a partially effective vaccine, it will make a difference. They're basing this mainly on things like phase 2 data, where there was a good immune response to these vaccines. And they didn't seem to have too many adverse events either. So based on phase 2, they're optimistic about phase 3. And the new technologies that are being used are compelling enough, to be honest. The RNA approach is new. The adjuvants they're using, GSK are giving out their adjuvant to many different vaccine candidates. These are new approaches. We failed to get vaccines to the common cold, but that was 25 years ago. We didn't have these new technologies. So that's why there's a degree of hope in this process, but we still don't know. We have to wait till those phase 3 trials are carefully done and safety is carefully monitored. And then, of course, just finally on the vaccine idea, there's BCG. This is a review I did just a couple of months ago now, actually, where BCG, which is the TB vaccine, has been known for decades to cause a kind of a non-specific innate response through epigenetic reprogramming of monocytes, and you'll boost cytokine production upon reinfection. It's been shown to work in RSV, influenza, and HSV too. So will it work in SARS-CoV-2? We don't know. There's a big phase 3 trial running at the moment on this. The idea here is it'll be a bridge to the vaccine. So BCG, if a new pathogen emerges, give BCG to cause this innate response, and then the vaccine might come along later. Let's wait and see what that BCG trial tells us. If it gives any kind of a signal at all, that's reasonably, compelling, that'll be a really good add-on. We think it's very safe. And now finally, the anti-inflammatory part. So as we know, this is effectively an inflammatory disease, COVID-19. Lungs get badly damaged. And it's all about macrophages and lymphocytes and cytokines. We all know these words now, don't we? The general public even know these. So this is a standard ARDS type scenario. You do see slight differences. It's a coagulopathy, seems a bit more intense with this one. I've worked on LPS and sepsis for years. If you inject LPS into lungs, you'll get a similar phenotype to COVID-19. So there's overlap with other sort of, infections that cause this response, a septic type response, you might say. And that gives us ideas of new drugs. Now, of course, the antivirals could work here. And there's remdesivir, New England Journal trial. A paper was published yesterday, New England Journal. That is showing effects. I mean, it's doing something. It's not gangbusters. It isn't a magic bullet by any means, but it is delaying time in hospital. There's a hint of an effect on fatality at the more severe end, so it's something. They've tried some of these anti-HIV drugs and they haven't really worked very well. We've gone off chloroquine, of course. I mean, there is some evidence, though, that chloroquine given early can have antiviral effects. So I think it's gone from this disease, though. And here's all the cytokines. Tocilizumab is targeting IL-6. That has failed in truth, and some of those trials didn't give a good readout. I think GM-CSF is very interesting. That may show more promise. And then here's all the antibody-based approaches, be they nanobodies and single chain antibodies and all sorts, and even convalescent plasma. That's sort of touted as well. That's a concern. I'm not sure that's going to work. There's a couple of issues about that, but then you never know. These things are all being explored as we speak. And then remember, as you would all know as well, the death rate's going down dramatically. This is the case all over the world. Here's a good piece of data that I dug out. We're comparing April to May, April 17th to June rather, that kind of period. And look at this, there's a drop in the death rate at every age group. This is tremendous, and there's all kinds of reasons for this. It's just clinical care has changed a bit. Use of anticoagulants is helping. And of course, this drug, dexamethasone, this is the first drug, I think, that clearly showed a benefit in truth. I worked on this as part of my PhD many years ago now. There's the New England Journal paper. Here we have mortality, dex versus usual care. You can see a 20-30% decrease in the death rate. This is the severe end if they're on ventilation, of course. But still, this is something, another weapon to use. So we're seeing hints of this now. This will get better. We'll see more therapeutic options emerge that target the inflammatory process. They could well be used in combination with antivirals as well. And then if we put antibodies in the mix, you can see a scenario where this no longer becomes a life-threatening disease in hospitals. Now, of course, the dream is to stop people getting to hospital with the vaccine, but still, this is okay. This is very helpful, we think. And very finally, then, my own work, we're working on them. I've worked on inflammation for 35 years now, various inflammatory pathways, highly complicated. And our most recent stuff is on a metabolite called itaconate. This is made by macrophages, and it's made from glucose through Krebs Cycle, if you can believe it. I know you all love Krebs Cycle. And there it is. And now we're testing it in COVID-19. Our evidence suggests that it was a potent anti-inflammatory natural metabolite. We do work on sepsis. This is in 2018. We published this. Here's a septic model in mice. Give them itaconate. You can extend life in sepsis. Other labs saw that and began trying against viruses. There was a paper about a year ago now in Immunity on Zika virus, where here we see lethality from Zika, and then they gave itaconate and they could protect the mice from dying from Zika virus. And they had a little diagram in their paper. Here's itaconate being sprayed on the virus. That looks a bit like COVID too, doesn't it? So itaconate could be another option to limit this and prevent lethality at one level. And then another really interesting study that gave me even more confidence was there was a multi-omic study, actually it was, but the metabolome in COVID-19 patients. And in their discussion, without me telling them, look at this, itaconic acid, a macrophage metabolite, was reduced with severity. Now remember, we think it's anti-inflammatory. So that if you have less of it, you might get more severe disease. That inspired us to do experiments. Here's a bit of data. This is SARS-CoV-2 growing in lung epithelial cells. The cells die by the truckload in this model. Give them itaconate and they live. Look at this, there's an itaconate dose response here. So we can stop the epithelial cells dying from COVID-19. And that could be antiviral or it could be anti-inflammatory, and you're maintaining the cell integrity there. We're doing lots of experiments on this at the moment. And we're very excited. As you would all know, anybody in research, you can't predict anything, but still we're going full steam ahead with our itaconic work now as a possible treatment, in fact. And we have derivatives that could be active as new medications. Again, it's a new idea. We got a lot of novelty, a lot of innovation here to beat this. And let me just finish where we're going to go next. Now, Devi Sridhar, one of the best epidemiologists in the world, I think she's fantastic. And I agree with everything she says. She said, look, this is about 3 weeks ago. There's three options now. Let the virus go. That's herd immunity. Thousands die. Lockdown and release will destroy the economy and society. Test, trace, isolate, soft distancing. She's pressing for that. And we got to keep saying that, especially the measures that we're all trying to follow with regard to physical distancing. Very important. And remember, our worries are, as case numbers go up, that translates almost mathematically into hospitalisation and fatality. Here's a great US-based study where look at this. There's the daily cases going up, and there's hospitalisation. Now, this big jump here was because there were older people back in April. But look, now it beautifully parallels. This is now going up here. It's slightly behind it, it must be said. But we know in the UK, other studies are showing increased case numbers means increased hospitalisation. So we got to get these numbers down to stop that happening. It's extremely important. And then we might think about a possible scenario for the next 6 to 12 months. It may be that immunity might begin to build up. Let's hope so. Even though it's not in everybody, there'll be some degree of immunity in the population. That'll help get the R number down. A vaccine, of course, helps that tremendously, even if it's only partially effective. So this double sort of thing where it's natural immunity combined with the vaccine has to help us. Therapies, I've said already, will improve. Great hope. Antibody-based therapies, I think, will be the first. They can be given as a prophylactic, and in fact, the notion of shielding has to be to do with medical-based shielding. We can't possibly shield the vulnerable. Anybody who advocates shield the vulnerable, let the virus out in the community, has to be questioned, because let's say you're in a household with an elderly relative or a middle-aged relative who's got heart disease on statins, or a teenager with diabetes. How do you protect those people? It seems impossible. So we do need a shield. The shield could be antibody-based. Obviously, the vaccine is a great way to shield. Or even remdesivir could be given as a prophylactic. So we're going to see more prophylactic approaches, I believe. Very importantly, with all these advances, even if they're imperfect, and let's say it's 6 months from now, the fear begins to go. Now, I'm talking about governments. They can take more risks, and the population fear level has to be got down, because the worst thing your immune system hates most of all is anxiety and fear. So I think we can be a bit optimistic that if we fast forward 6, 9, 12 months, some of these things will start to yield, and then fear begins to go, and ultimately we'll see a way out of this. I think that's the glass half full scenario, and it is based on what we're learning about the therapeutics and the vaccines that I just mentioned to you. That's scientifically based, that opinion. It isn't just sort of a wing and a prayer territory here. Now, of course, things can go wrong, and you have to be skeptical and sort of, somewhat, realistic about these things. But still, I think this is a reasonable scenario for the future. And remember, together we'll beat it, and we beat it using all the various things that we're doing, including social distancing and all the various measures that are in place. So I shall finish there, and thank you very much.